Welcome to Azad Bioacademy. I will just now come to the third stage. That means extracellular stage or peptidoglycan synthesis stage, or you can say cross-linking stage. You can consider this is the membrane, that is lipid bilayer, okay, and everything now will be happening outside of the membrane, that is within the periplasmic space. Okay, so you can think you already got a sugar, that is N-acetyl muramic acid, and which is attached with the N-acetyl glucosamine, the basic chain of the sugar chain, and you know that N-acetyl muramic acid will carry pentapeptide. First one is L-alanine, second one is D-glutamine, third one is, that is L-lysine, and fourth and fifth one are basically D-alanine and D-alanine. Okay, so this is the one unit. So another unit will come up by the flippase or bacterial. So that unit will attach here. That unit will attach here. So you can add here. So this is the N-acetyl muramic acid, and this is the N-acetyl glucosamine. Okay, so now another unit, that is N-acetyl glucosamine, then N-acetyl muramic acid, and N-acetyl glucosamine and N-acetyl muramic acid will add here by the forming bond here, that is the glycosidic bond. Okay, this is the glycosidic bond, and that bond will form by the particular enzyme. This name is, that is PBP, it's penicillin binding protein. You can also, it is also known as transpeptidase. Okay, and the transpeptidase or penicillin binding protein has several types. Okay, basically it is two types. One is high molecular weight PBP and low molecular weight PBP. High molecular weight PBP has class A and class B. So class A has two functions. One is transpeptidation, that is cross-linking, and another one is transglucosylation. Okay, that means it will form a glucosidic bond. Okay, this class A, and class B has one single function, that is cross-linking. So both class A and class B is responsible for causing cross-linking, but class A is also another specific function, that is glucoside bond formation. Okay, that means trans-glucosylation. And the low molecular weight PBP, that is transpeptidase enzyme, has some role, that is decarboxylation. You can say endopeptidase activity. That means it is basically responsible for remodeling or maintenance of the peptidoglycan layer. Okay, so it has some decarboxylation, that is cleavage activity, and embedding activity, that is remodeling activity of the peptidoglycan. Okay, so you can say remodeling and maintenance activity. So this is okay. So now both high molecular weight and low molecular weight combinedly work together. That means combinedly work to form a cross-linking and peptidoglycan formation. Basically, peptidoglycan formation, cross-linking formation, both enzyme works. So it has multiple number of transpeptidase activity, but in a single bacterium, that is single species, almost 6, 7, 8 transpeptidase enzyme will be available to form the peptidoglycan layer. Okay, so now here one PBP, that is transpeptidase enzyme, will help to form a bond between two N-acetyl glucosamine and N-acetylmuramic acid, glucosamine and N-acetylmuramic acid. And if you think it is happening here outside of the membrane, so where the enzyme will be there? Actually, this enzyme, the penicillin binding protein, that is transpeptidase enzyme, is basically attached to the membrane. Okay, and it will work from here to there. That means it has connection. But actually, for your easy understanding, I'm drawing here out just far away from the membrane, but it is very adjacent to the membrane, and it is you can think like it is like that enzyme there. And if it is work here, the enzyme is there. Okay, it is attached to the membrane for your kind information. So now this unit will also carry the pentapeptide. Actually, I'm sorry, I forgot to add the pentaglycine. So, for your easy understanding, I'm drawing here. That is the pentaglycine. Okay. So here it will also carry the pentaglycine. Okay. So now what will happen? Another unit, that is the basic unit of sugar chain, that is skeletal unit of sugar chain, that will carry the peptide, pentapeptide. And what will happen? Another layer will be there and it will make a cross link. So what will happen? Now you can draw here. This is just I'm drawing here. This is, you can say, now this is NAM, this is now, and there that is alanine, then glutamine, then and finally it will also carry pentaglycine. Okay, in that way, again another unit of sugar, and then another unit of sugar, and here will be there also here also it will carry the penta and sugar. So now just I am simply I will now try to make you clear about the cross-linking. So here the enzyme will work. That enzyme is transpeptidase. That transpeptidase will work on the fifth number of 4 and fifth number of amino acid, that is D-alanine dimer. Okay, this is the D-alanine dimer, this is the D-alanine dimer, and this is the D-alanine dimer. The structure of D-alanine dimer, between the D-alanine, carbonyl group is there. Okay, so this is carbonyl group there, and transpeptidase enzyme in the active functioning position, that active position of the enzyme will carry serine, and serine has OH group. That means if you think this is the enzyme, and here serine are there, and serine will carry the OH group, and here the carbonyl group. So now that enzyme will attach here. The enzyme will attach here. This is the enzyme will attach here. This enzyme is PBP, penicillin binding, that is transferase enzyme. And initially it will attract to each other by the hydrogen bond. Invisible hydrogen bond will attract to each other, and then it will form a bond between them. That is, you can say, acyl enzyme intermediate. So it will form a bond there by acylation between them, and it will form a acyl enzyme intermediate. Then what will happen? When it will form a acyl bond with the dipeptide, then what will happen? Sorry, the bond stage will be there. Okay, so then what will happen? Here, it is basically the high molecular weight PBP, and it is also attached with the low molecular weight PBP. That means combinedly it is working, that is transpeptidase. So what will happen now after binding with the dipeptide D-alanine, and it will form a acyl bond, and then the low molecular weight penicillin binding protein will cleave that fifth number of D-alanine. It will cut off, that means it will cleave. And by the cleavage of that fifth D-alanine, here energy will produce, and that energy will make that bond very strong. That is, covalently it will form esterification, that is acyl ester bond with the fourth D-alanine. and here that fifth DLNA is just gone. Okay, and energy will produce, and that energy will work to form this bond very strong, and it will facilitate to work with another pentapeptide. Then what will happen? It will just manage, that enzyme will manage to form a bond by the pentaglycine, will make a cross-link. That is glycine, 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 and glycine. So the bond will be between 3 to 4 linkage. Number 3 and 4th. One chain, another chain will be attached by the cross-linking. Okay, that is pentaglycine cross-linking. And you know the pentaglycine is Staphylococcus aureus carrying pentaglycine. So between them it will form a cross-link. So here also the pentaglycine will form with the 4th link. So in that way it will form a bond by pentaglycine. Okay. So which species, some species of bacteria like E. coli has no pentaglycine. So in that case what will happen? In that case actually that will happen like actually those are very adjacent to each other, and it will form a direct bond between amino group of that third number of carbon, amino acid, and here carbonyl group, and it will form a bond, covalent bond, that is amide bond. Amide covalent bond will form, and it will attach directly by 3-4 linkages. Most of the bacteria, the bond is available between 3 to 4. Some bacteria 4 to 3, some bacteria 2 to 4 may be possible, but most of the cases 3 to 4 linkages. So two types of cross-linking, actually I am trying to focus on that two types of cross-linking. One is 3 to 4 cross-linking by pentaglycine, and 3 to 4 direct cross-linking by covalent bond, basically amide bond, you can say peptide bond. Okay, so this is the cross-linking by the enzyme help of transpeptidase, or you can also say penicillin binding protein. Okay, so this is all about the third phase of the peptidoglycan. So you can consider this is the peptidoglycan layer, and you can consider it the cell wall, because the cell wall, basic component of the cell wall is peptidoglycan. So this is the peptidoglycan synthesis or cell wall synthesis. So it has 3 stages. One is your cytoplasmic stage, that is initiation. Second one is the membrane stage, that is elongation and transmigration. Third one is your extracellular stage or peptidoglycan synthesis, or you can say cross-linking stage.